Alright, welcome back to Biology Rough Cuts. So today we're going to be talking about, let's see here, where is my screen? So today we're going to be talking about pseudogenes and a really interesting function of pseudogenes as a mutational mechanism. Now, there's a couple things I want to cover. First of all, this has been known for a while, but it's almost never talked about. It's fascinating, and the language people use to describe it is almost amusing because it's obviously a mechanism, but nobody really wants to call it one. So you kind of have to dig through interesting language in order to find out what's actually going on. So, so basically, what's happening is that pseudogenes, since they're not actual genes, they're basically what they're doing is they're a repository of alternate information for other genes. I mean, that's not the only function. There's a lot of things pseudogenes do, and actually, we're just barely finding out what pseudogene function is. But one of the things that they do is they provide a repository of information for express genes. So basically, if you think about a gene, if it needs alternate configurations, well, those alternate configurations can be stored in a pseudogene and then recombined back into the gene. So again, most people have the idea that mutation is haphazard, but actually it's actually very controlled. And so you see these mutations are very controlled where it's literally copying segments from a specific pseudogene to a specific gene to provide for additional diversity. So one of the ways, so this paper, this is Balakarev and Ayala, and Francisco Ayala has actually been involved in a lot of the research on function of pseudogenes. And this particular paper is really interesting. It has a lot of things, but the one that I wanted to focus on is the pseudogenes are reservoirs for generating genetic diversity. This is absolutely fascinating. So especially, I'm going to talk about the antigenic variations. All right, so it says pseudogenes act as a reservoir of sequences that recombine with their functional paralogous sequences and thus generate antigenic diversity. Gene conversion events have been reported in several bacteria. Let's see here. Bacteria of the genus Borrelia generate antigenic diversity of their coat proteins through recombination from a tandem array of silent partial pseudogene cassettes into a telomeric expression site on a linear plasmid. So basically what's happening is that you have, so the outer coat protein, which is what bacteria need to survive in different environments. Okay, so this is literally the thing that needs to be changed a lot. It's got, so the main gene protein, so it has a sequence, but then it has a set of pseudogenes. Okay, and what happened is that as it needs variation in its outer coat, these these pieces of a pseudogene get copied in. Now, it might not be as directed as, say, oh, I need it, and now copy it in. It may be to the effect that these are known good alternatives that are being copied in, and maybe that is randomized to some extent. But the fact is that the origin and the destination are well regulated, so that these mutational events are biased towards function. So this is one thing. There's a difference between random with respect to selection and random with respect to function, and this is an important distinction that I think needs to be made more often. So it's I don't know. Perhaps it knows which ones to recombine with, but the data that we have so far just says that it knows what to copy from and where to copy it to generate a biologically sensible outcome. And so anyway, so again, antigenic variation is generated in Anaplasma marginale by recombination of pseudogenes into the functional expression site. So yet another organism where you have pseudogenes that are being, and this is for antigenic variation. So that's the what the antibodies attach to. So it's variation because it's trying to evade immunity. Pseudogenes are also a source of genetic diversity in gonococcus. These bacteria generate palin variants that allow evasion of the host immune system. So a partial pilin sequence is introduced into the expression locus to form a complete pilin gene. All right, so their pili are basically are, um, are basically altered by the pseudogenes. Pilin phase variation can also occur by deletions between direct repeats internal to the expressed pilin gene or between a pilin gene and an adjacent silent pseudogene. All right. So even more, antigenic variation of the surface proteins of mycoplasma is achieved by recombination between multiple pseudogenes and a pressed VLHA gene. The multiple pseudogenes serve as a repertoire for the creation of VLHA sequence diversity, probably by site-specific recombination, with recombination between the single complete VLHA gene and one of the multiple partial copies, creating new VLHA gene variants. So anyway, so the expressed genes are likely to be chimeric with sequences derived from several overlapping pseudogenes. The discrete boundaries of sequence similarity observed for the pseudogenes strongly suggest a site-specific mechanism of gene conversion. Right, so gene conversion often happens when you have sequences that are very similar in both the origin and the destination. Recombination between VSG genes and pseudogenes has been suggested as a mechanism by which antigenic diversity is generated in Trypanosoma equiperdum, as well as in T. cruzi genome by recombination between surface protein pseudogenes. Again, so all of these are genes which organisms need continual variation on. And so what they have is these pseudogenes which are providing those alternatives. And so this next one has nothing to do with evolution, but it's also interesting. The human olfactory receptor pseudogenes may be important for the generation and maintenance of receptor diversity. All right. So intensive intergenic gene conversion has been revealed for this multi-gene family that leads to segment shuffling in the odor biting site. All right, so the ability to have different smells, and basically similar to VDJ recombination in the immune system, which we'll talk about. It's not really VDJ recombination is not a excessively important one from an evolutionary perspective, but nonetheless it provides some information on ways that the organism can affect its own DNA. So anyway, so these pseudogenes are super important for generating diversity. So I also want to point to a paper, if I can find it. There it is. So this is a focusing homologous recombination, pilin antigenic variation in the pathogen Neisseria. Right. So I want to show this both because it's interesting data and the way that it talks about it. So anyway, so some pathogenic microbes utilize homologous recombination to generate antigenic variability in targets of immune surveillance. All right, so basically places where the immune system might hit it, it's going to generate variability. These specialized systems rely on the cellular recombination machinery to catalyze dedicated high-frequency reactions that provide extensive diversity in the genes encoding surface antigens. Okay, so they're not saying that it's for that. They're just saying it happens to do that, right? This is the kind of the language that you'll oftentimes find. A description of the specific mechanisms that allow unusually high rates of recombination without unusually high rates of recombination without deleterious effects on the genome in the well-characterized pilot antigen variation system of Neisseria gonorrhea and Neisseria meningitis is presented. All right, so unusually high rates of combination. All right, so this is the thing that's happening a lot without deleterious effects. So we're having recombination without problems. Okay, so in other words, it's saying that this is happening over and over and over again, and it's not causing any issues. Well, in fact, it's not only not causing any problems, it's generating the antigenic diversity that's needed for it to survive. All right, so it's not, but the thing is that it's not like if you think about the concept of random mutation. Well, a random mutation will generate bad stuff and good stuff. Well, this is a system that's only generating good stuff, or at least biologically significant stuff. So if we go through here, all right, so pilin antigenic variation occurs as a non-reciprocal DNA recombination between one of multiple PIL-S silent storage loci and the pilin expression locus PIL-E. All right, so it's a non-reciprocal DNA recombination. That means it goes one way. It goes from PIL-S to PIL-E. Okay, so it's basically saying we've got these silent pseudogenes, and the main expression site, we're not going the other way. We're saving these PIL-S genes because they're important. We need the sequences. We're not coming from the produced gene back to the saving storage area. We're only going from the saving storage area to the gene. Okay, so it's a non-reciprocal DNA recombination. Let's see here. I was thinking there's some other things that we need to look at here. So this goes into some of the molecular factors of the system, which is interesting, but I'm not sure that they actually have the complete system fully done yet. This is more of a suggestion of where they're looking. Let's see here. We talked about VSG already. Let's see here. So another thing that's really interesting is that in this section they talk about it actually requires, they have very specific recognition sites to show it where to do the modification, because if you modify, there's a lot of these base pairs that if you modify them, so the way that it says is what the effect is on pilin antigenic variation. Okay, so what they're meaning is that the directed mutation does not happen. So this one says it had no effect, but there's let's see here. When individually mutated, absolutely inhibited pilin antigen. Okay, so 11 GC base pairs, when they are individually mutated, absolutely inhibited pilin antigenic variation. So what they mean is that this sequence is super important for it to be able to know that it can mutate here. Okay, so this is so in order for the organism to recognize this as a place that it can create variation from, it has to have a sequence that points it out to it. So there's literally a detector sequence that that the cell is able to use to tell where the recombination should occur, and if there's mutations there, it won't know to do recombination there. So anyway, it's just super fascinating. this is a great area of study. Unfortunately, it's not better known. Um, so I hope that so this is and this is only, I mean, there's probably other ways that pseudogenes are useful evolutionarily, but this is and they're all and they've been finding all sorts of interesting uses of pseudogenes generally. But I just want to point out using them as stored, um, basically stored diversity for later incorporation into the expression sites of genes that need variation is an extremely important and extremely interesting um, uh, usage of pseudogenes.